Hi guys, I'm Andrew. I'm going to be presenting with Caylee and Victoria on the paper of the Chromothriptic cure of WHIM Syndrome by McDermott et al.
To get us started, I'm going to describe Chromothripsis to everyone. So this is quite literally a genetic event in which a chromosome shatters. The chromosome attempts to piece itself back together, most likely by non-homologous end-joining, but this results in massive rearrangements and deletions. The resulting chromosome may rearrange in a way that puts an oncogene in a promoter region, or so that a critical gene is missing. Chromothripsis is typically seen in cancer cells, but we're going to present a case in which the outcome is quite different. We're going to change gears here a bit, and I'm going to talk about WHIM syndrome. WHIM syndrome is an autosomal dominant immunodeficiency disease caused by a mutation in the CXCR4 receptor. The CXCR4 receptor is responsible for production and distribution of white blood cells in the blood and bone marrow. The phenotype of the disease is actually described by the name of the syndrome itself, which is an acronym for its primary manifestations. WHIM stands for warts, hypogammaglobulinemia, infections, and myelocathexis. The disruption of white blood cell production and distribution results in a compromised immune system in these individuals. This is why recurrent infections are an issue. The immunodeficiency leads to HPV infections, which causes warts that cannot be handled using standard medical treatments. Their HPV infection can also lead to cancer. Hypogammaglobulinemia refers to low levels of immunoglobulin G, or IgGs. IgGs are antibodies responsible for memory in the immune system, and these low levels contribute to the recurrent infections seen in these patients. Finally, myelocathexis occurs when neutrophil migration from the bone marrow, where they're produced, to the bloodstream is defective. This leads to neutropenia, or low levels of neutrophils in the bloodstream. So as you can see, this mutation mainly affects immune responses and pathways. Currently, the primary methods of treatment focus on supplementing IgGs. This helps to reduce the number of infections seen in patients. HPV vaccinations are also highly recommended, but they need to be re-administered periodically as the patient's adaptive immune system is compromised. Certain medications, as well as bone marrow and hematopoietic stem cell transplants, are under investigation as possible treatments. So like I said previously, WHIM syndrome occurs due to an autosomal dominant mutation in the CXCR4 gene, leading to a gain of function. Under normal circumstances, CXCR4 is expressed by most white blood cells and responds to one constitutively expressed ligand, CXCL12. This pathway mediates hematopoietic stem cells, or HSCs, retention in bone marrow by supporting dormancy in HSCs and preventing differentiation into myeloid cell types. The mutation causes a disruption in the negative regulatory elements of the gene and results in an increase in signaling. The process that prevents myeloid differentiation into things like neutrophils and basophils is occurring at abnormally high levels in these patients, leaving HSCs dormant in the bone marrow rather than differentiating and egressing to the blood, and leading to lower observed levels of neutrophils in the bloodstream. I want to stop here for a moment and explain the process by which HSCs differentiate to different white blood cell types. HSCs are the precursor to all blood cell lineages, and they mature and differentiate in the bone marrow as depicted on the previous slide. Here we'll be focusing specifically on white blood cells, or leukocytes, though. As HSCs divide, some will become myeloid progenitor cells and others will become lymphoid progenitor cells. Once this differentiation has occurred, each cell is destined down its path. It cannot regress back to an HSC. Myeloid progenitor cells can further differentiate into several cell types, as you can see here, but the most important for our study are neutrophils and monocytes. Lymphoid progenitor cells will go on to become natural killer cells, T cells, or B cells. Just as a quick refresher for everyone, neutrophils activate inflammatory responses and play a vital role in frontline response to pathogen. Monocytes contribute to the ability to attack pathogens and also facilitate healing and repair. B cells recognize foreign invaders on the outside of infected cells and tag them. T cells then recognize and attack those infected cells. Knowing how all these cells function, it's simple to see how those with WHIM syndrome present with the symptoms that we've talked about. Now that we've had a brief refresher, Andrew is going to get into the specifics with this patient's clinical presentations.
Okay, guys, I'm really excited to tell you about this story. I mean, this is just an incredible, crazy event that happened, and I'm just going to talk about how incredibly lucky also this patient is. So let's jump right into it. So here in figure A, we have the pedigree with the index patient, and if you remember, that's indicated with the arrow, and her 3 daughters. This index patient is a white female who presented with WHIM syndrome at age 9 and is one of the first described with the disease. So WHIM09 also has 3 daughters, 2 affected, 1 daughter, which is WHIM10 at age 21, and another daughter, WHIM11 at age 23. Both of the daughters have had a history of all the symptoms that Caylee talked about with the recurrent infections, warts, and etc. But I mean, here's just the crazy thing, like mysteriously, there's like a spontaneous and complete remission of the confluent warts in a patient WHIM09. You know, just absolutely crazy. So yeah, and here in figure B, it clearly shows the stark difference with WHIM09's remission hands versus her daughters, right? You can see it, just the nasty warts in 11, and there's none in 09. So after looking at the patient's at WHIM09's medical history, none of her parents or siblings had WHIM, so the WHIM mutation must have occurred de novo in the patient 09 with an autosomal dominant transmission to 2 of her daughters. So the whole point of WHIM09 coming back to the doctors and researchers was trying to figure out, like, why in the world did she randomly recover from this nasty disease? So that's what we're all going to get into today. So next, they looked at the potential mechanisms for how this clinical remission occurred. So they looked at the blood cell counts in WHIM09. So she initially had pretty severe neutropenia, low neutrophil count, and monocytopenia, which is low monocyte count. But again, both resolved spontaneously over time. So here in figure C, there's that little arrow, which indicates when WHIM09 had a splenectomy, removal of the spleen at age 9. And then the dotted horizontal lines indicate the normal range for each cell type, right? So you can also see in the x-axis there's a nice little break because WHIM09 stopped coming in to get tested and for observations, and then she spontaneously improved, so she came back again to see if they could figure out what in the world happened to her. So as you can see in all these graphs, the white blood cell count, absolute neutrophil count, absolute monocyte count, and absolute lymphocyte count all inconsistently increased over time. Okay, so next they genotyped whole blood DNA from the family members using a restriction fragment length polymorphism assay for the CXCR4 mutation. The daughters tested positive for the mutation, when 10 and 11, and the uninfected husband and third daughter both tested negative, right? So that's not that surprising. The mutation was not detected by direct sequencing of the whole blood cell from DNA from WHIM09, right? So just to recap, in figure F, they used Sanger sequencing for the whole blood DNA, and they were looking for nucleotides in the position of around 1,000, which is the vertical line where the CXCR4 mutation is. Then in figure G, they did some more Sanger sequencing from DNA from the blood neutrophils, that's the top line, a buccal swab, aka cheek swab, right, and the skin fibroblasts. This shows that WHIM09 is heterozygous for the CXCR4 mutation and is a somatic genetic mosaic, right? So just really confusing. And as you can imagine, very confusing for the researchers too, right? So then they try to look at maybe, just maybe, if there's like a genetic reversion involved or maybe something else, right? So just this crazy story continues. Okay, so here's where the Chromothripsis aspect comes in. The researchers were interested in maybe a reversion or deletion in the WHIM09 leukocytes in the nucleotide that determines the CXCR4 allele occurred. So in this figure A, they did a karyotype of WHIM09's bone marrow cells. They noticed that one copy of the CXCR4 was significantly shorter than its normal homologue. One of the copies were acrocentric, when the other copy was normally submetacentric. And here's a figure on the right that shows the difference in case you forgot. And all the other chromosomes were normal. I should also note that for the banding pattern of the weird chromosome 2, it looked like that there had been some inversions also. Then they did a microarray and found specifically that the abnormal chromosome 2 was 35 megabases shorter because of seven large deletions. So overall, WHIM09 is hemizygous for the CXCR4 from the loss of the mutation. By whole genome sequencing, they saw that the chromosome was made from 18 remaining pieces in random order and in random orientation, which is pretty characteristic of Chromothripsis, right? So in this figure B is a Circos plot, which I'm pretty sure we've seen before in other presentations. Basically, large pieces of DNA were missing. A total of 163 genes were lost. I mean, that is insane, and there were no deleterious effects. Absolutely crazy, right? And then the 18 remaining pieces were rearranged in random order. Where these pieces moved are represented by the colored lines, and the inner circle is the copy number variation from their microarray from before. So from the paired end sequencing analysis, the connections where the fragments move matches where the copy number variation quickly jumps and changes from 2 to 1. You can see that in the inner circle. So lastly, they used an ideogram, which is not pictured on this slide, from the karyotype that they used before, where they looked at pairs of the intact homologous chromosomes in the nucleus, and they determined specifically what novel fusion genes were made. So all the new genes that were created weren't expressed because they didn't have a promoter or a transcription initiation site, which is just absolutely crazy and, again, incredibly lucky for this WHIM09 patient. So the most likely explanation for WHIM09's improvement was the haploinsufficiency from the Chromothripsis. Okay, so here these two figures are representing the myeloid mosaicism from the Chromothripsis. Since all the neutrophils were uniformly chromothriptic from the whole genome sequencing, the PCR, and then the cytogenetics, this probably meant that at least some of the HSCs, hematopoietic stem cells, also had to be chromothriptic. So here in figure B, they looked at this CD34 leukocytes from the peripheral blood and bone marrow that lacked the CXCR4 allele. And then moving on to figure D, they looked at the CLPs, the CMPs, and the GMPs, with all the abbreviations here on the right. And the CMPs and the GMPs lacked the CXCR4 allele, which is consistent with figure B, right? But the CLPs still had the allele. They did all this using PCR on Chromothripsis specific primers. So basically, as a summary, in figure F, all the mature myeloid cell types lack the CXCR4 allele in red, where all the mature lymphoid cells were heterozygous for the WHIM allele in green. So now we know that this crazy Chromothripsis event did indeed occur. Now Victoria is going to talk about the stem cell engraftment and bone marrow transplantation in mice.
So now that Andrew has covered that a chromothriptic event occurred in the single hematopoietic stem cell of WHIM9, the researchers wanted to figure out how this single chromothriptic hematopoietic stem cell was able to outcompete all of the other mutant hematopoietic stem cells that was still in WHIM9's bone marrow and blood. And so to do this, they used a CD45 Congenic Mice model, and they modeled bone marrow transplantation. And so I want to give a brief reminder about what congenic mice are, and specifically what CD45 congenic mice are. So congenic mice are inbred mice. These have been inbred for multiple generations until they have the exact same genetic background, except for at a single allele. And in CD45 congenic mice, as the name implies, this single allele is the CD45 allele, which is also called protein tyrosine phosphatase receptor. And the reason that this is used widely in bone marrow transplantation is that the CD45 protein is expressed on all hematopoietic cells except for mature erythrocytes and platelets. And so these congenic mice have two different alleles, CD45.1 and CD45.2. And the utility of these alleles is that they can be targeted with specific antibodies that are fluorescently tagged. And so here I have them highlighted in their colors that will be seen on the next few slides. And so the reason that this is important for bone marrow transplantation is that it allows for the tracking of hematopoietic stem cells from donor mice and from recipient mice. And also another huge benefit of the fact that these congenic mice have the same genetic background is that there's no risk of immunological rejection of the donor cells. So here's a schematic of how this bone marrow transplantation occurs in an experiment. And so here we have labeled those CD45 congenic mice. We see donor 1 expresses CD45.2, which is fluorescently tagged in red, and donor 2 is heterozygous for CD45.1 and CD45.2, which are fluorescently labeled with green and red respectively. And then the recipient is labeled with CD45.1, which is tagged with green fluorescence. And so first, the donor bone marrow cells have to be harvested from the bone marrow surgically. And then these cells are sorted in a fluorescently associated cell sorter, or FACS, so that you just get the hematopoietic stem cells. And this occurs for donor 1 and donor 2. And then their hematopoietic stem cells are mixed together in equal proportions. And meanwhile, before these are transplanted into the recipient has to be irradiated because we want to kill off all the recipient's cells, because the whole point is to see how the donor cells engraft and proliferate and repopulate. So we have to kill off all the recipient cells to see how the donor cells do in the recipient. So after the recipient has been irradiated, the donor cells are injected through the tail vein in equal proportions. Now, with this first bone marrow transplantation experiment, what the researchers wanted to figure out was how did that one Chromothriptic hematopoietic stem cell outcompete all of the other mutant stem cells in the bone marrow? And so really the question they're asking here is CXCR4 haploinsufficiency sufficient for advantageous repopulation of the bone marrow in WHIM syndrome? And so to the left, we have our experimental design. And this is showing that we have one donor cell, which is haploinsufficient for CXCR4. And this was generated by knocking out this CXCR4 gene with Cre-Lox. And then this mouse was bred onto the CD45.2 background. We also have a donor mouse that's representing WHIM syndrome. And this was an established mice model, which was then bred on a CD45.1 and CD45.2 heterozygous background. Of course, we have to irradiate the hematopoietic stem cells of our recipient mouse, which is bred on a CD45.1 background. And so using the fluorescence that we can generate from these different hematopoietic markers, we can analyze the input of the experiment and also the results using flow cytometry. So I'm briefly going to describe how to read these flow plots. So on the y-axis, we have our CD45.2 marker, and on the x-axis, we have our CD45.1 marker. And so if we were looking for our haploinsufficient donor, we would expect them to cluster near the top left corner of the flow plot because they're expressing high CD45.2 and they shouldn't be expressing any CD45.1. We would expect our second donor, which is heterozygous for both CD45.1 and CD45.2, to cluster more towards the middle of this flow plot because it's highly expressing both CD45.2 and CD45.1. And then we don't expect to see any CD45.1 cells, which would be clustering more towards the bottom right, because those have been lethally irradiated. So in this first flow plot, what we're seeing is that hematopoietic stem cells from the haploinsufficient mouse and from the WHIM mouse model were harvested in equal proportions and injected in equal proportions, and those are 44.6% and 44.4% respectively. And in the second plot, we're seeing what the proportion of stem cells measured from the bone marrow, measured from the blood at day 105, what their origins are. And so again, we see the blood cells derived from the haploinsufficient mouse clustering more towards the top left, and we can see that 50% of the cells that were gathered from the recipient were actually from the haploinsufficient mouse, while only 5% of those cells were from the WHIM syndrome mouse. And then we also do see that some of the recipient's own cells are recovering, even though they were supposedly all irradiated, but we see about 1% of the recipient's own cells recovering. So what this shows us is that those haploinsufficient hematopoietic cells really do have an advantage in repopulation. Here we see a very similar slide to the last slide, but here the researchers are asking a slightly different question. And this time the question is, in general, is CXCR4 haploinsufficiency advantageous for repopulation of the bone marrow after transplantation? So here we're comparing haploinsufficient mice to wild-type mice. And so this really isn't related to WHIM syndrome anymore, but just looking to see what this CXCR4 haploinsufficiency means in bone marrow transplantation in general. And so again, we have this experimental design to the left where we're just showing that the haploinsufficient mouse is marked with CD45.2, while the wild-type mice is marked with CD45.1. And again, they're being transplanted into irradiated mice. The flow plots show that the hematopoietic stem cells from both donor mice, the haploinsufficient mouse and the wild-type mouse, were injected in fairly equal proportion, 35% to 48%. So we do see that the wild-type mouse might have a slight advantage to begin with their 48% of the cells. And the next flow plot shows that at day 303, when blood was drawn from the recipient mouse, actually the great majority of the hematopoietic cells were derived from the haploinsufficient mouse, with 33.5% being derived from the haploinsufficient mouse and only about 6% being derived from the wild-type mouse.
Based on this study, McDermott and colleagues were able to determine that chromothripsis, a form of chromosome instability, actually resulted in the benefit of clinical cure of disease in WHIM9. They found that the disease allele could no longer be detected in HSCs, neutrophils, or monocytes in this patient, which were severely affected before when WHIM9 was initially studied. These findings allow scientists to say that she was in complete remission and effectively cured. They were also able to make new deductions regarding HPV. Because the patient remained lymphopenic but did not show HPV symptoms, it's likely that myeloid-derived immune cells play a more important role in fighting HPV. Finally, shockingly, they also determined that haploinsufficiency for CXCR4 is actually advantageous in wild-type individuals, not just mutants. These determinations all add up to allow us to deduce that chromothripsis occurred in an HSC in WHIM9, giving the affected myeloid lineage a selection advantage over the CXCR4 mutant myeloid lineage. The implications of this study suggest that gene editing could be a successful future endeavor for those studying treatments for WHIM syndrome. We've seen how the absence of the mutant allele results in a return to normal function, and this is promising for the future. This study also provides important data on how we can improve bone marrow transplantation. Further studies should be done to determine how we can capitalize on the advantage of haploinsufficiency seen here.
So the point that I want to leave you all with is that WHIM9 won the genetic lottery. What are the odds that this chromothriptic event in a single hematopoietic stem cell would lead to clinical remission? First of all, chromothripsis is normally this hugely damaging event that occurs in a cell. And even in this hematopoietic stem cell of WHIM9, there were fusion genes created that just so happened to not be expressed. There were 163 other genes that were deleted, but these genes must not have either been expressed in the white blood cell, or they just did not suffer from haploinsufficiency even though they were on an autosomal chromosome. And then again, what are the odds that this chromothriptic event happened in a hematopoietic stem cell, which was the affected tissue type? And this was just an amazing event that the NIH was able to document from start to finish, because WHIM9 was the first patient that was described to have WHIM syndrome, and then also the first patient described to have clinically recovered from WHIM syndrome. So really, it's just an incredible story. And here are our references. Thank you so much for watching, and we hope that you enjoyed this presentation. We look forward to questions in class.